we are still working on our phenomenon on how organisms repair an injury. And so we're specifically going to focus on the cell membrane, which is found in both the animal and plant cell. And we're going to look at the how it functions, and that's going to show you how it begins to help repair injuries at the cellular level. And this is going to happen through osmosis and diffusion, which require little to no energy of the cell and of the organism. So it's transportation across the cell membrane. It's moving from molecules of water, sugar, proteins, anything that can get into and out of the cell. So specifically with diffusion, it's most likely sugar or small molecules that don't require energy for the cell to take in. So the membrane in a cell allows these molecules to come in and out kind of as they please. But diffusion is the movement of molecules from an area of high concentration to an area of low concentration. High concentration means there's a lot, say, of sugar on one side of the cell membrane, and there's a low amount of sugar on the other side. So the sugar that's on the high concentration side will move to the side of the cell membrane that has a low amount of sugar. Osmosis is a type of diffusion, but it's specific to water only, and it's water going across this semi-permeable membrane, which is what we call the cell membrane. It's semi-permeable because it allows some things in and out very easily, but other things, such as larger molecules of protein, it would require some other energy and another form of transportation. So again, osmosis is just water going from an area of high concentration to an area of low concentration, and diffusion is any other molecule that might be able to move across the cell membrane without using energy. And molecules move across the cell membrane. So sugar and water, they're going to move across the cell membrane, and that's to keep the cell functioning. Plant cells need a lot more water because remember, they're just like drinking it out of the soil, and we can get water when we want, so it doesn't mean that our cells need to store as much water, whereas plants are going to need that storage capacity, so they're going to bring in as much water into each of their cells as possible. In this little diagram here, this is the cell membrane. Okay, it's permeable, and it's showing diffusion. It's showing there's a lot of, say, water molecules on the other side, on the outside of the cell. And then it's showing you over time these water molecules start to move through the semi-permeable cell membrane, and they end up inside the cell. And then they're going to end up so that there's the same amount of water on the inside of the cell as there is on the outside of the cell. And then again, some are going to move into the cell while others move out, because remember, the cell also has to get rid of waste products. Okay, so this is kind of what we call an osmosis or diffusion kind of problem. You'll see that we have our cell membrane here. So this is inside the cell. And we have a couple water droplets, okay? It's labeling those as water. And this is outside the cell. Well, in order for osmosis to occur, we know it goes from an area of high concentration. So high concentration means there's a lot of water droplets, to an area of low concentration. There's only 2 water droplets inside the cell. So that means that we're going to have water move across the membrane this way into the cell. Okay? So that there are equal number of water droplets inside and equal number outside. So that means 2 of these water droplets would have to move into the cell, so that there would be 4 water droplets in here and 4 water droplets out there. So that means these 2 water droplets are going to move into the cell. And that would give what we call equilibrium. So equal number of water droplets on the inside and equal number of water on the outside. Okay? Because remember, osmosis is water going across the cell membrane. So it's like a semi-permeable membrane. So water is going to flow from an area of high concentration to an area of low concentration. Now let's look at this second problem. Oops. You'll see that this one I kind of included a plant cell. So you have inside the cell and outside the cell, you have the cell wall and then the cell membrane. And then you have 6 water droplets on the inside of the cell and 4 on the outside, okay? So then water is going to have to go across the membrane this way, okay? And then we would only need, in order to make it equal, 1 water droplet is going to move out, so that there would be 5 water droplets, or 5 water droplets on the outside, 5 on the inside, to make it complete. So again, this is osmosis, which is the diffusion of water across a cell membrane, okay? And we want to make sure that the amount of water on the outside of the cell equals the amount of water on the inside of the cell. And this is going to help cells carry out their function, which is why they get rid of certain things across their membrane. Pay attention to the next couple of videos that you're going to watch, because it's me performing an investigation or a model that shows you how osmosis works and that it really does work. And next week, you're going to have to come up with your own model to show osmosis and or diffusion. Dialysis tubing, which you see right here, is what we're going to use in this experiment or demonstration that I'm going to do with you, so that you can understand how things flow across a cell membrane, so into and out of a cell, such as waste or food or water specifically. And so dialysis tubing is what we call a semi-permeable membrane. It allows different molecules like sugar to flow in and out of it. Okay, so we're going to use that as part of our experiment. So it is kind of see-through, so it's kind of hard to see. And then down here you'll see I have my 3 beakers, which you're hard to see, but this is our control, which is going to have no sugar in this water. And then this is going to have 1 tablespoon of sugar. I haven't done it yet, but I will. And this has 4 tablespoons of sugar in it, okay? And each dialysis tube, I've knotted at the bottom. You can sort of see the knots in there. And then I clipped to the side of the beaker. And inside each dialysis tubing is water, just plain old tap water. And then we're going to measure them over time to see how much they weigh, if they've changed in mass, to see if they've gained any sugar water, okay? And we're going to use a scale and electric balance to do that. So now I've put sugar in both the 1 tablespoon and 4 tablespoon beakers. You notice that there's sugar actually at the bottom of each one because it has more sugar than the water can hold, but I'll keep stirring them over time. So in about 20 minutes, I'll take the mass again. I've already taken the mass for 0 minutes, which is what we do before our experiment starts. So in about 20 minutes, I'll take the mass. This is what they look like after about 100 minutes. So not much has really changed at the eye level, but when you look at my data table, then you'll see that the mass in each of the tubes has changed slightly, although my control has changed, and I'm not really sure why, but I think I might have some ideas, so I will talk about that a little later. So this is an iodine solution. It's just water with some iodine in it. And here I have dialysis tubing filled with starch solution. And when iodine touches a starch solution, this is cornstarch, and so when iodine touches a starch solution, it turns blue. So I put the dialysis tubing in with the iodine. And the theory is that the dialysis tubing is a semi-permeable membrane, and iodine molecules can pass through. If they can pass through, then they should turn the inside of the dialysis tubing blue, because iodine detects the presence of a starch. So you should see at the bottom that the dialysis tubing is starting to turn a blue color. The longer we wait, the more of the tubing will begin to turn blue, and you can see there is actually some blue on the inside.